Hello, and thank you for watching the first segment of the physiology of the reproductive systems. In this first segment, we'll cover slides 1 through 58, and in segment 2, we'll cover slides 59 through 112. In this first segment, we will cover the first 3 points here. We'll talk about the production of gametes, and that applies to both the female and the male reproductive systems. Gametes are our sex cells, they're egg and sperm. And then we'll focus for a little while on the male reproductive system. In segment 2, we'll pick up at point number 4 here, and we'll focus mostly on the female reproductive system. Meiosis plus fertilization forever. They're a match made in heaven. No, not quite. So these two biological processes have opposing effects. Meiosis is a type of cell division that divides the genetic complement in half. So each resulting cell has half the full complement or the full set of chromosomes. And then fertilization, when egg and sperm meet, that's the biological process that restores the full genetic complement. As each egg and sperm have half the number of chromosomes during fertilization, those two halves come together to restore the full genetic complement. So most of our body cells are somatic cells. Somatic cells, remember, soma means body. Somatic cells are not involved in the formation of egg or sperm, meaning their specific genetic material, those chromosomes inside somatic cells, will never be passed on to offspring. So which cells here are somatic cells? And we've talked about most of these cells. We see familiar cells in the kidney, we see hepatocytes in the liver. We've not really talked too much about bone cells. We see lung cells in alveolar sacs, neurons in the brain. We see red and white blood cells and platelets. We see cardiomyocytes, heart muscle cells. We see skeletal muscle cells or smooth muscle cells. And we see, it looks like those are acinar cells in the pancreas making pancreatic juice. So do any of these cells contribute their genetic material to egg or sperm? And remember, egg and sperm contribute their genetic material to the formation of new offspring. And the answer is no. None of the cells shown here contribute genetic material to egg or sperm. And so they're somatic cells. And these somatic cells have the full genetic complement. And what that means, at least for our species in humans, it means 46 chromosomes organized as 23 pairs. Just like you can imagine 23 pairs of socks where each sock has its match. 23 pairs of socks would give us 46 individual socks. And so we have 46 chromosomes arranged as 23 homologous pairs. And when we have our full genetic complement, as our somatic cells do, we call that the diploid condition, or 2N, where di means 2. So 2 of each of the 23 chromosomes are present. So what's a chromosome? A chromosome is a package of DNA. And this DNA, as we can see here, this DNA is tightly wound around proteins called histones. So the DNA is coiled very tightly and in a highly organized fashion around these histones, and then these units of DNA plus histones, those units are coiled upon each other again. So it's a coil of coils of coils into this discrete unit known as a chromosome. This is a photograph, a photomicrograph, a photograph taken through a microscope. And what's shown here is a person's karyotype. These are all their chromosomes. And a karyotype shows the chromosomes that a specific individual has. So these chromosomes came from one person. And the way we make a karyotype is by starting with a blood sample. And we need to access the chromosomes which are present inside the cell nucleus. And I hope you remember that red blood cells don't have nuclei, so they're not useful for this process, but white blood cells do. And we can separate the red cells from the white cells. And once we have the white cells, we can apply signaling molecules that cause those white blood cells to go into mitosis, a very common form of cell division that occurs in our body. And we'll talk a lot more about mitosis in a bit. But when these cells start to undergo mitosis, their genetic material condenses. These chromosomes coil very tightly. So now we have these pretty easy to manipulate packages or units of genetic material. And so we induce these white blood cells to begin the process of cell division in order to get their chromosomes to be all tight and dense. And then before they have an opportunity to finish cell division, we pop open the cells, we split open the nucleus, and the nucleus spills its chromosomes everywhere. And then we add a stain. And that stain is a molecule that binds to other molecules. And that stain gives us the characteristic banding patterns that we see on each of these chromosomes. And then the chromosomes are photographed. They're all in a jumble, much like if you took 23 pairs of socks out of the dryer and threw them on the bed. Those socks wouldn't be matched up. They'd be in a jumble. And if you took a photograph of that jumble of socks and then you manipulated that photograph to line up the matching pairs of socks. So this photograph has been manipulated so that the matching pairs of chromosomes line up. And we see that there are 23 pairs of homologous chromosomes here, where homologous means the two chromosomes have the same size and shape, they have the same banding pattern, and they carry the same type of genetic material. So we have 23 pairs of homologous chromosomes shown here. Pair 1 through 22, those are known as autosomes because they have nothing to do with determining the biological sex of a person. The last pair, shown on the bottom right, those are called sex chromosomes, the X chromosome and the Y chromosome. They're called sex chromosomes because they do determine the biological sex of a person. So imagine this is your karyotype. For each pair of chromosomes, you inherited one of these chromosomes from your biological mother. It was present in the egg before it was fertilized. And the other chromosome you inherited from your biological father. It was present in the sperm prior to fertilization. And then during fertilization, egg meets sperm, their nuclei merge, and we restore this full complement of genetic material. This is why you have 23 pairs of homologous chromosomes. Biological sex can be determined by looking at an individual's karyotype. Remember, the X and the Y are sex chromosomes, and females, biological females, will present with 2 X chromosomes, and a biological male will normally, typically, present with 1 X and 1 Y chromosome. Notice that this karyotype comes from a biological male, and also notice that the X and the Y chromosomes are not homologous, meaning they have different sizes, they have different banding patterns, and they carry different genetic material. Biological sex is determined by chromosomes, and it's different than gender. Gender is a social facet of an individual, how one integrates into society. And it's related to biological sex. For many people, their biological sex matches their gender. But for many other people, their biological sex does not match their gender. And then, not shown here, but we also can use a karyotype to see what are known as chromosome abnormalities. So lots of people in the world, their karyotype looks different than this. They have an extra chromosome, or they're missing a chromosome, or one of the chromosomes is broken, for example. And with the sex chromosomes, it's possible, for example, to have only 1 X chromosome. There's no Y and there's no complementary X. That's known as Turner syndrome, just to have a single X chromosome. Another example of a chromosome abnormality that involves the sex chromosomes is Klinefelter syndrome. And in Klinefelter syndrome, the person's karyotype shows a single X chromosome and 2 Y chromosomes. So we're going to talk about the production of gametes. Gametes means egg and sperm, or sex cells, as sometimes they're called. And these cells are special. They're special because they contain half the number of chromosomes in a somatic cell. So each egg and each sperm contains 23 chromosomes, and they're 23 unpaired chromosomes. So if we think about that, those socks that we pulled out of your dryer earlier and we put them on the bed, what if we selected one sock from each of those 23 pairs of socks? Though that represents the haploid state, or 1N. So here we can compare side by side the chromosomes that occur inside of a generic somatic cell versus a gamete cell or a sex cell. And so it would be too complicated if this diagram actually had 23 pairs in the somatic cell and 23 individual chromosomes or unpaired chromosomes in the gamete cell. So maybe this comes from a different organism. Let's see, how many chromosome pairs does this organism have? 1, 2, 3, 4, 5, 6, 7, 8, 9. So this comes from some organism that has 9 different chromosomes in its genome. And the somatic cell shown here has 9 complementary pairs, 9 homologous chromosomes, 9 paired chromosomes. And the gamete cell, it's either an egg cell or a sperm cell, has 1 of each of those 9 chromosomes. So the somatic cell is diploid, or 2N, and the gamete cell is haploid, or 1N. So when we have these two haploid cells, an egg and a sperm, during a process known as fertilization, these two cells merge and their nuclei fuse. And so the 23 individual chromosomes that are in the egg meet the 23 individual chromosomes that are in the sperm, and in the resulting embryo, the developing embryo, we find in the embryo cells, we find 46 chromosomes, and they are in 23 pairs. It's as if every one of your unmatched socks found its pair. So there are 2 types of cell division in the body, and part of the last slide involved both of these types of cell division, mitosis and meiosis. So at various points along the semester, we have encountered mitosis. Mitosis is vastly more common in our body. It's happening, you're making millions of new cells every second by mitosis. And meiosis only comes up in humans. Meiosis is only important, it's only relevant in the reproductive system because it's how egg and sperm are formed. So let's talk about mitosis. This is the type of cell division that's happening in your skin to make new skin cells, in your bone marrow to make new blood cells. It's happening in the lining of your GI tract to replace those mucosal epithelial cells. So mitosis is happening lots of places in your body right now, and it's how one parent cell essentially clones itself to give rise to 2, and the terminology is to call them daughter cells, even though there's no sex or gender involved here. So during mitosis, a somatic body cell, in this case, divides where one cell becomes 2, and each of the 2 resulting daughter cells have 46 chromosomes. And again, these 46 chromosomes are in 23 homologous pairs. So 23 pairs of chromosomes, that's 46 individual chromosomes. Mitosis is happening all over your body, all the time. It occurs millions of times every second. So if you've ever injured yourself and watched yourself heal, that certainly involves mitosis, where damaged tissue cells can no longer function, and so healthy tissue cells divide by mitosis to replace the damaged tissue cells. Hemopoiesis, I hope you remember that refers to the formation of new blood cells, and that occurs in red bone marrow. I also hope you remember that red blood cells in particular they're short-lived, they have short lifespans, they only live for a few months. And so red blood cells are constantly being taken out of circulation, and new red blood cells are being put into circulation. And the way new red blood cells are produced is through mitosis. And then keratinocytes. We have not talked too much this semester about skin, but the outer layer of skin, the most superficial layer of skin, is called your epidermis, and we are constantly shedding dead skin cells as a way of preventing microbes from colonizing our surface. If we're constantly shedding our surface, then those microbes can't really get a foothold normally. And so in order to replace those shed keratinocytes, those keratinocytes deeper in our epidermis, they must be dividing by mitosis, and they are right now. And then we recently covered the digestive system and that epithelial lining of the GI tract. It's part of the mucosa. There's an epithelial lining that's in contact with the food in your mouth and your esophagus and your stomach and so on. Those cells are shed and replaced constantly. And so those cells are undergoing mitosis right now. And then during fetal development, after fertilization, that egg and that sperm come together to produce a new single cell known as a zygote. And that zygote has 46 chromosomes. It has the full genetic complement. Well, how do we go from a single cell to a fetus? Mitosis happens so that all of the resulting cells have that same full genetic complement. Meiosis is a different type of cell division. And in animals, and humans are animals, it only happens in the gonads, ovaries or testes. And this type of cell division is used in the animal body to cut in half the full genetic complement. So if our somatic cells have 46 chromosomes, egg and sperm have 23. Well, how do we get a cell that has 23 chromosomes? The answer is meiosis. So unlike mitosis, meiosis has two successive cell divisions, appropriately called meiosis 1 and meiosis 2. So the whole process of meiosis involves a first division followed by a second division. And so one parent cell that undergoes meiosis gives rise to 4 daughter cells. And again, this has nothing to do with sex or gender, it's just the terminology. We call these 4 resulting cells daughter cells, and each of these daughter cells has half the full genetic complement. So in humans, each of these daughter cells is going to have 23 chromosomes. And importantly, this is not cloning. In mitosis, which somatic cells undergo, in mitosis, the daughter cells are genetically identical to each other, and they are genetically identical to what was the original parent cell. In meiosis, the 4 daughter cells are all genetically unique. They are not identical to each other. And this is why siblings that share both parents, this is why siblings are not genetically identical, because no egg is genetically identical to another egg. No sperm is genetically identical to another sperm. So here's a schematic layout of meiosis. Meiosis begins with a diploid parent cell. Diploid in humans means that there are 46 chromosomes present. Then that diploid cell undergoes meiosis 1, the first division, and gives rise to 2 daughter cells. And then each of those daughter cells undergoes meiosis 2, the second division. And the outcome of meiosis is 4 daughter cells that each have half the genetic complements of the parent cell. So each of these daughter cells has 23 unpaired chromosomes, And each of these daughter cells are genetically unique. So here we can do a side-by-side comparison of mitosis, shown on the left, and meiosis, shown on the right. And in this course, we are not going through the phases of cell division. Perhaps in a previous course, you have examined the phases of cell division: prophase, metaphase, anaphase, telophase, and then cytokinesis, or the separation of that one cell into two new cells. We're not covering those phases, but I wanted to remind you of them here because you've probably encountered them in a previous biology course. What I'd like to point out, what is super relevant for us, are highlighted in those two boxes. So in the box on the left, we see the outcome of mitosis. Mitosis, that's the far more common type of cell division. It's happening in your skin, in your bone marrow, many other places in your body right now. So the outcome of mitosis is that for every one parent cell, mitosis yields or gives rise to two daughter cells, and each of these daughter cells is diploid, just like the parent cell was diploid. There are 46 chromosomes present, and the two daughter cells are clones. They're genetically identical to the parent cell. And then if we look at the right panel, we see meiosis, and meiosis only occurs in your gonads, in the testes or the ovaries. And there are two successive divisions. So on the right side, the right panel, the top half of that in sort of the light yellow background, that's meiosis 1. That's the first division. And then below that, with the darker yellow background, that's meiosis 2. That's the second division. And the outcomes, the important outcomes of meiosis for us, are shown in the box on the right. For every one parent cell, meiosis yields 4 daughter cells. We're going to have to modify that in a bit when we talk about how eggs are formed. So this is true for sperm. What eggs do is a little different. So meiosis results in 4 daughter cells. Each of these daughter cells has 23 chromosomes, so they're haploid. And none of these daughter cells are genetically identical to each other. And of course, they only have 23 chromosomes, so they can't possibly be genetically identical to the parent chromosome. Now we move into examining the male reproductive system, which is quite different from the female reproductive system. And so far, all semester long, biological sex didn't matter. Cardiovascular systems, digestive systems, urinary systems, a little bit in urinary systems. But when we come to reproductive systems, we're really studying two different organ systems, the male reproductive system and the female reproductive system. So let me give you an overview of the male reproductive system. So if we start with the testis is where spermatogenesis occurs, and spermatogenesis is the formation of sperm. So meiosis that we were just discussing in the male body, meiosis occurs in the testes. And the testes, they're paired. There are 2 of them, a right testis and a left testis. And these 2 testes are housed in really a sac of skin called the scrotum. And then each testis has an epididymis. And the epididymis is where mature sperm are housed prior to ejaculation. And then for much of the rest of the male reproductive system, we can follow what's known as the ejaculatory pathway. So the pathway that semen travels during ejaculation. And from the epididymis, peristaltic contractions, these wave-like muscular contractions, will propel sperm in fluid through the ductus deferens, also known as the vas deferens. And that vas deferens loops over the ureter, shown here. And then we meet up with our first accessory sex gland called the seminal vesicle. And again, all of this is paired. This happens on the right side of the body as well as the left side of the body. So the seminal vesicle is one of the accessory sex glands, and it secretes some of the components of semen, the fluid components. And so as the sperm and the fluid pass through the seminal vesicle, they meet up with secretions from the, pass through the vas deferens, they meet up with secretions from the seminal vesicle and enter this short little tube here called the ejaculatory duct. The ejaculatory duct passes through the prostate gland. The prostate gland is another accessory sex organ. It contributes different components to semen. And so as those cells, that sperm and that fluid passes through the ejaculatory duct, it gains more fluid from the prostate gland, and then that leads into a little bit of the prostatic urethra. That's the portion of the male urethra that passes through the prostate gland, and then into the membranous urethra. The membranous urethra is that segment of urethra that's neither in the prostate gland nor in the penis. So there's this short segment of urethra called the membranous urethra. And then the urethra changes names again. Once it's in the penis, it's called the penile urethra, or sometimes it's called the spongy urethra, for reasons that we'll talk about in a minute. So as this semen passes through the spongy urethra, it eventually reaches the external urethral orifice, and that's the passageway out of the body. So the male reproductive system and urinary system overlap in that the urethra is a common passageway for both urine and semen, and that's not true in the female reproductive system. In the female reproductive system, the urinary system and the reproductive system are completely separated. What else can we see here that is important for us? We can see some of the anatomy of the penis. I just mentioned the external urethral orifice. The head of the penis is known as the glans penis. The foreskin, which is this loose skin that is in the United States, it's pretty common to remove that shortly after birth in a procedure known as, what's that called? A procedure known as circumcision. The foreskin or the prepuce is removed. And then the little lip around the head of the penis is known as the corona. Nothing to do with the virus. And then internally, inside the penis, we see different erectile tissues that we'll talk about in a little bit. The corpus spongiosum is one of those erectile tissues. It's spongy. It has holes. It's sponge-like in appearance. And the corpus spongiosum surrounds the penile urethra, which is why the penile urethra is sometimes called the spongy urethra, because it's surrounded by the corpus spongiosum. And then the other erectile tissue belongs in the corpora cavernosum, and these are two rods of erectile tissue. We'll take a different view of this in a moment. But both the corpora cavernosum and the corpus spongiosum during erection, they fill with blood. And so the penis is really a hydrostatic pump. As that hydrostatic pressure inside the penis increases, it becomes erect. And we'll talk more about that in a little bit. We also have our third accessory sex gland here, known as the bulbourethral glands. Again, they're paired, and they contribute a little bit towards semen, and they are actually more important prior to ejaculation. They make pre-semen, or maybe you know it as pre-cum, which helps neutralize any acidic residues from urine that's left in the urethra. Urine, it's quite common for urine to have a low pH, and that low pH is not so bueno. It's not so good for the sperm that are coming down the pipe. And so secretions from the bulbourethral gland that occur during arousal, they line the urethra and help raise the pH to make it a more hospitable environment for the sperm that are about to pass through. And then secretions from the bulbourethral gland also help to lubricate the tip of the penis for insertion. All right. So some details of spermatogenesis. I've walked you through the basic mechanics of meiosis in sort of a generic form. Here's the basics of meiosis. Well, how does meiosis happen specifically in the human testes or the animal testes. So genesis means generation of, right? So it refers to the process by which sperm are formed. And spermatogenesis doesn't occur in babies or young children. It starts at the onset of puberty, and that age is different for different people. Sometime between roughly 8 and 12 years old is when spermatogenesis begins. And hormones, signaling molecules, released by the anterior pituitary are responsible for this change in behavior of testicular tissues. Why is it that cells in the testes go from being fairly dormant to becoming fairly active during puberty? And it's under the influence of these hormones from the anterior pituitary. And these hormones are required to stimulate both the production of testosterone as well as spermatogenesis. In the reproductive system, we'll be talking a lot about steroid hormones. So steroids are lipids. They're a category of lipid, just like a phospholipid is a lipid. Steroids are lipids, and they're hormones, they're signaling molecules, and they're fat soluble. So in the reproductive system, we'll be talking about groups of signaling molecules, groups of steroid hormones known as androgens and estrogens. And androgens, by definition, are masculinizing sex hormones. And we see a variety of androgens here. The most well-known androgen is testosterone, but there are other androgens shown here like androstenedione and dihydrotestosterone, and they have similar molecular structures, and one is often easily converted into the other, and they act on similar receptor subtypes. Estrogens are, by definition, feminizing sex hormones. And there is no one individual molecule called estrogen, but when we talk about estrogen, we're really referring to a group of closely related signaling molecules like estrone and estradiol. So we have androgens and estrogens, and all bodies have both, regardless of biological sex. If we were to take a blood sample from you right now, we would find both androgens and estrogens in your blood. We would just find them in different proportions given your biological sex. So in the male body, we tend to find higher circulating levels of androgens, and in the female body, we tend to find higher circulating levels of estrogen. And the primary source of these sex hormones are the gonads. So in the female body, the source of estrogens is the ovaries, and in the male body, the source of testosterone and other androgens is the testes. But all bodies have androgens and estrogens, and these hormones are also produced by the adrenal glands, specifically the adrenal cortex. That's the outer layer of the adrenal gland. And we talked about the adrenal cortex in the context of the urinary system because it's the source of aldosterone. And I'll remind you that aldosterone is secreted as part of the RAA pathway, the renin-angiotensin-aldosterone pathway. And that's engaged as part of blood volume, blood pressure homeostasis. So in that context, we talked a little bit about the adrenal cortex previously. The adrenal cortex is coming up here because in both the male and the female body, it is the source of steroid or sex hormone synthesis. So the androgens that are in my blood, I'm biologically female as far as I know, I'm biologically female. The androgens that are coursing through my blood right now, they were produced in my adrenal cortex. And in my sweetie, as far as we know, he's biologically male. The source of estrogens that are circulating in his blood right now is the adrenal cortex. So I definitely don't want you to memorize, I do not want you to memorize the pathways of steroid production. In addition to what we just talked about, I would like you to recognize that cholesterol, this molecule over here, is the starting ingredient for the production of all steroid hormones. And cholesterol is an important molecule. It's gotten a bad rap in previous years, and some people would engage in a low cholesterol diet in an attempt to ward off heart disease. That's coming under question now. We're not so sure about the relationship between cholesterol, dietary cholesterol, and heart disease. Cholesterol is important biologically for two reasons. Number one, it's a component of all your cell membranes. So the plasma membrane of every cell in your body includes cholesterol, and that cholesterol helps keep that membrane fluid or supple or pliable. It's bendy. Another reason why cholesterol is important is it's the starting ingredient for all of your steroid hormones. And in the reproductive systems, we're focused on androgens and estrogens. And cholesterol, it's okay if you don't eat it because your liver will make cholesterol. The reproductive system functions of the testes, the production of sperm and the production of testosterone, begin at the onset of puberty. And what triggers the change in behavior in the testes are hormones that are produced by the hypothalamus and the anterior pituitary. And then this continues throughout the rest of a man's life. There's no such thing as menopause in the body of a biological male for the most part. So this process that we're about to take a look at begins at puberty and continues for the duration of a man's life. There is a lot to be said in this diagram. So let me start by pointing out the three different sources of hormones that we see here. The first is the hypothalamus, the second is the anterior pituitary, and the third are these Leydig cells, which are also known as interstitial cells. So the hypothalamus is traditionally thought of as part of the brain. The anterior pituitary is one half of the pituitary gland. The pituitary gland is known as the master gland because it secretes hormones that in turn control several other endocrine structures, including the testis. And then the Leydig cells are the source of testosterone produced by the testes. We will also eventually talk about Sertoli cells. Sertoli cells do not produce hormones, but they do produce androgen binding protein. And if you recall, testosterone is a steroid hormone, which means it's lipid soluble, which means it needs a chaperone protein in order to circulate in blood. And that chaperone protein makes testosterone more soluble in blood plasma. It also prevents individual packets of testosterone from merging with each other in blood. So it keeps the packets of testosterone separated, so to speak, and makes testosterone more easily transported in blood. And this is androgen binding protein, and it's produced by the Sertoli cells. Sertoli cells will come up again when we talk more about spermatogenesis, or how sperm are formed. So the hypothalamus, part of the brain, secretes a hormone. It's also an endocrine structure. So now we're talking clearly about the neuroendocrine system. The hypothalamus produces a hormone called gonadotropin releasing hormone, or GnRH, shown here. Gonadotropin releasing hormone exerts its effect on the anterior pituitary. And the cells in the anterior pituitary respond to GnRH by secreting more of these two hormones, FSH and LH. FSH stands for follicle stimulating hormone. It was initially discovered and named after examining ovaries, but it is also active and plays an important role in the function of the testis. So FSH stands for follicle stimulating hormone, and LH stands for luteinizing hormone. And these two hormones secreted by the anterior pituitary exert effects on cells in the testes. And in this diagram, the effects of FSH are somewhat distinguished from the effects of LH, but for us, for our purposes, we'll always talk about these two hormones together. Collectively, FSH and LH stimulate spermatogenesis in the testes and testosterone production in the testes. So spermatogenesis in the testes is shown here, and we'll talk more about that later. So these are cells that are in the process of undergoing meiosis as well as spermiation. We'll talk about that later. And then our second important cell type here are the Sertoli cells. They are bizarre and super cool cells. So maybe I can outline one Sertoli cell here. I'm doing a terrible job of it. As far as hormonally, we are concerned with Sertoli cells because they make androgen binding protein, which is that chaperone protein for testosterone. And under the influence of FSH and LH, these Leydig cells, which are outside of the seminiferous tubules, we'll talk about that a little bit later. These Leydig cells, or these interstitial cells, produce testosterone. And this testosterone is responsible for male secondary sex characteristics. So think about what happens to the typical male body at puberty. We'll talk about those in a little bit. And testosterone is also required for the onset of spermatogenesis. So we have a hormone secreted by the hypothalamus, gonadotropin releasing hormone, which triggers the secretion of FSH and LH from the anterior pituitary, which triggers the production of testosterone by these Leydig cells. It also triggers the production of androgen binding protein from these Sertoli cells. So circulating levels of testosterone will increase. Notice that there's a feedback mechanism here. Let me switch to a different color. Notice that there's a feedback mechanism here where circulating levels of testosterone are detected by both the anterior pituitary as well as the hypothalamus. And they have, as shown by these little minus signs here, circulating levels of testosterone can contribute to a negative feedback system. So when circulating levels of testosterone are high, that feeds back to both the hypothalamus and the anterior pituitary, and each of these structures will respectively decrease their secretion of GnRH and their secretion of FSH and LH, and that will help bring down circulating levels of testosterone. So the secretion of pituitary hormones and the resulting secretion of testosterone are regulated by feedback mechanisms, as we were just talking about. So testosterone here inhibits the secretion of gonadotropin releasing hormone here, and it also inhibits the secretion of FSH and LH. Here's a question. How would the hypothalamus respond to a decrease in plasma testosterone? So now plasma testosterone is lower than normal. How does the hypothalamus respond? Well, it would respond by increasing the production of gonadotropin releasing hormone. That in turn, GnRH stimulates the production of FSH and LH from the anterior pituitary. And with increased circulating levels of FSH and LH from the anterior pituitary, that will lead to an increase in the production of testosterone by these Leydig cells. And of course, the level of testosterone in the blood is constantly being monitored by both the hypothalamus and the anterior pituitary gland. So this is a typical negative feedback system. So let's take a look at the overall functions of the male reproductive system. Starting with spermatogenesis. Spermatogenesis is the production of sperm, and it occurs inside these structures known as seminiferous tubules. So just like in the kidney, we had renal tubules, these microscopic tubes, these little tiny tubes. We have seminiferous tubules in the testes, these little tiny microscopic tubes. And inside of the tube is where sperm are formed. And then not really well represented here, but there are cells outside of the tube. So maybe you can envision them here around the tubes. These are called interstitial cells. They're also known as Leydig cells, and those are the cells that secrete testosterone. Here we're looking at a photomicrograph of testis, and we can see transverse sections through seminiferous tubules. So this is one seminiferous tubule here. Here's a neighboring seminiferous tubule here. And then in between the seminiferous tubules are patches of cells. These are the interstitial cells or the Leydig cells, and this is where testosterone is produced. Testosterone is required for the production of sperm, so there's some local signaling that happens. These Leydig cells produce testosterone, and yes, that testosterone gets into the blood, and that testosterone influences many tissues around the body, as we'll talk about, but the tissue that we're looking at right here, testosterone is required for spermatogenesis, so it influences what's happening inside this seminiferous tubule. And so these cells here, shown here, I'm coloring them in, these are cells that are engaged in various stages of spermatogenesis. And we'll look at the stages of spermatogenesis in a little bit. So what we're looking at here in this green shaded region are Sertoli cells plus cells engaged in spermatogenesis. And spermatogenesis occurs from superficial to deep. So the cells that are in the earliest stages of spermatogenesis are closer to the exterior, to the surface of the tubule, and spermatogenesis proceeds as we move toward the interior. And so here, all of these little lines that you see here, those are all sperm tails, they're all flagella. And then once these cells are mature, they will swim through the seminiferous tubules into the epididymis, where they will be stored prior to ejaculation. So let's take a look at these cells that are engaged in spermatogenesis. So we were just looking at a cross section through the testis, and then we can highlight one seminiferous tubule. So it's a transverse section through a tube, like a hose, and we're going to zoom in on one portion of a seminiferous tubule. So here's the exterior surface of the tubule, and here's the lumen of the tubule down here. And so we have these cells here that are in various stages of spermatogenesis. And we can start with a spermatogonium. A spermatogonium is a diploid cell. There are 46 chromosomes present in a spermatogonium. And then moving toward the interior, we pass through the stages of spermatogenesis, including a primary spermatocyte, a secondary spermatocyte, spermatids, and then these spermatids undergo a streamlining process to become sperm. So here we have to overlay our understanding of both mitosis and meiosis onto these cells in the testes. So before we talked sort of in generic terms about, well, what's mitosis and what's meiosis, and now we can apply those to these cells specifically in the testes. So here, this is the exterior coating around a seminiferous tubule. And then we have spermatogonia. Spermatogonia, you can think of them as sperm stem cells. Spermatogonia exist to give rise to primary spermatocytes. And they do this following puberty. They'll do this for the duration of a man's life. And spermatogonia are diploid cells. Notice that there's a 2N inside this cell, so that indicates it has 2 copies of each chromosome, 2 copies of the 23 pairs, giving rise to 46 chromosomes. And the spermatogonium undergoes mitosis. So notice that both daughter cells here are also diploid. These are genetic clones to the original parent cell. And one of these daughter cells will remain a spermatogonium. The other daughter cell differentiates to become what's known as a primary spermatocyte. And this primary spermatocyte will undergo meiosis to produce sperm. This other daughter cell up here that remains a spermatogonium, that lives on to repeat the process. So it just divides by mitosis. One of the daughter cells goes on to become sperm. The other daughter cell lives on to repeat the process of mitosis. All right, so here we have this primary spermatocyte. It's diploid. It's got 46 chromosomes, and it undergoes the first meiotic division. So during meiosis 1, one primary spermatocyte gives rise to 2, notice that we see 2 here, 2 secondary spermatocytes. So that's the first meiotic division. Each secondary spermatocyte undergoes meiosis II, which is the second meiotic division, and each one of these cells gives rise to two spermatozoa or two spermatids. I'm not really differentiating between the two right now. Let's talk about spermatids. So following meiosis II, we have four spermatids. These four spermatids don't really look like sperm yet. And in order to become functional sperm, they need to undergo spermiogenesis. And so in this process here, moving from a spermatid to a sperm, these cells differentiate, and part of their differentiation process is to develop a flagellum, this long tail of the sperm that you see here. Another part of this differentiation process is to shed most of the cell's cytosol, most of the cell's organelles. And so we end up with a sperm that's the smallest cell in the human body. It's essentially DNA with a propeller. We'll take a closer look at the anatomy of a sperm in just a little bit. Here's a question for you. What role does mitosis play in spermatogenesis? It plays an important role. Mitosis is shown here. Mitosis gives us this constant supply of primary spermatocytes, right? When a spermatogonium undergoes mitosis, one of those daughter cells will become a primary spermatocyte. That primary spermatocyte then undergoes meiosis to give rise to sperm. Sertoli cells. Sertoli cells play a few important functions in the testes. We've already talked about Sertoli cells being the source of androgen binding protein, ABP, that chaperone protein that travels with testosterone. Testosterone is a lipid-soluble hormone, so it needs a chaperone protein, and that chaperone protein is produced by Sertoli cells. Sertoli cells play an important function in spermatogenesis. They form what's known as the blood-testis barrier. And this is a protective barrier to prevent or to protect the sperm from whatever might be circulating around in our blood. For most of human evolutionary history, we didn't have agriculture. And so our ancestors were hunter-gatherers, and most of the organisms that we eat or we ate historically, those organisms produce their own protective molecules that are often toxins in our body. So we're eating a variety of plants, a variety of fungi, algae in our historical diet, and I'm speaking about us as a species, so globally. And then some of those food sources would produce toxins that have the capacity to be mutagens. They could induce mutation, and we want to protect our germline cells. Those are the cells that give rise to egg and sperm. And so in order to protect those germline cells in the male body, we have what's known as a blood-testis barrier. And that's made up of the Sertoli cells. The Sertoli cells only expose the developing sperm to materials that pass through the Sertoli cell body. So on this artistic representation, again, here's the barrier, the exterior surface of a seminiferous tubule. I hope you recognize that these are the cells engaged in spermatogenesis. So we start with our spermatogonium, which gives rise to a primary spermatocyte. A secondary spermatocyte gives rise to four spermatids, and these four spermatids will undergo differentiation to produce sperm. Here is a Sertoli cell. They have really cool shapes. They almost look like jigsaw puzzle pieces to me. I'll try to outline the plasma membrane of one Sertoli cell. And so you can see that the Sertoli cell sort of envelops the developing spermatogonia spermatocytes. Here's the nucleus of this one Sertoli cell. And not really shown here particularly well, but we'll take a look in the next image. These neighboring Sertoli cells, they have tight junctions connecting them to each other. And I hope you recall that tight junctions form a leak-resistant connection between cells. So here's a blood vessel. There are no blood vessels inside the seminiferous tubules, but there are cells, and these cells clearly need oxygen and nutrients. These cells give rise to wastes, and these wastes need to be carted away. So the oxygen and nutrients diffuse through this basement membrane here and into the seminiferous tubule, where the Sertoli cells act as a barrier. So in order to get to, let's say, a secondary spermatocyte, the oxygen and nutrients need to pass through the body of a Sertoli cell, and they just can't leak randomly. And this is a protective barrier. I found this diagram on the internet. I'm not sure who the author is, but I like it. Shown here, we see two different Sertoli cells, and they're labeled Sertoli cell 1 and Sertoli cell 2. We also see the exterior coating around a seminiferous tubule. That's the basement membrane. We can see the nuclei, the nucleus of each of these Sertoli cells. And then we also see these cells that are in various stages of spermatogenesis. We see the primary spermatocyte, secondary spermatocyte. Each secondary spermatocyte gives rise to 4 spermatids. And then that process of spermiogenesis is happening here as a spermatid, as a spermatid develops a flagellum and sheds a lot of its cytoplasm and becomes the streamlined cell that you recognize as a sperm. So the author has also included tight junctions here between neighboring Sertoli cells. And in order for material to reach these developing sperm cells, those materials, the nutrients, the gases, need to, let me change colors here. Let's go to yellow and see how that works. Those nutrients need to pass through the body of a Sertoli cell in order to reach a developing sperm cell. This is the blood testis barrier. It's recently been reported in a letter to JAMA that genetic material from the SARS-CoV-2 virus, the virus that's responsible for COVID-19, it was recently detected in the semen of many men. Maybe their sample size was somewhere around 30, perhaps 40. And these men had all tested positive for the virus, and they were in various stages of infection or recovery. So they were either in the stage of acute infection or they had recently recovered from COVID-19. And they produced a semen sample, and in the semen, genetic material from the virus was detected. We don't know if these were full infectious virus particles, so we don't know if the virus could potentially be transmitted sexually, or if these are just pieces of RNA, the genetic material from the virus. We do know that the genetic material was detected. We don't know if it was infectious or not. And so the question is, well, how does that viral genetic material get into the semen? We have a blood testis barrier that should prevent virus particles from getting into the semen. So remember, there are many access points into semen. It doesn't have to occur in the lumen of a seminiferous tubule. But these mature sperm will migrate to the epididymis, and then during ejaculation, these mature sperm will be propelled through the ejaculatory pathway. And so it remains to be determined, number one, if the virus material is actually infectious or if it's just pieces of viral RNA. And number two, how did that viral genetic material get into the semen? As a straight woman who doesn't want children, this is my least favorite cell type in the body. Sperm can be aptly described as DNA with a propeller. They are the smallest cells in the human body. They're streamlined. They have a torpedo-like shape. And these are the 3 pieces of a sperm. There's the head. The head includes the nucleus. This is where we find 23 unpaired chromosomes. And then the acrosome. The acrosome is part of the head. The acrosome includes vesicles. So you remember that vesicles are storage sacs. And inside these vesicles are storage sacs that are exocytosed, and that releases enzymes that are important during fertilization. It helps the sperm penetrate into the egg. So there's the head, there's the midpiece. This midpiece of a sperm includes many different mitochondria. What's the basic function of the mitochondria? Why would a sperm have all these mitochondria and pretty much no other organelles? The mitochondria produce ATP. That's right. Mitochondria produce ATP. And then we have our third piece of the sperm. This is the flagellum. It's a whip-like tail. In order for this flagellum to undulate, to beat, to spiral around, that requires ATP. And so the mitochondria that are in the midpiece produce the energy that allow the flagellum to move. And that movement of the flagellum produces this swimming motion, this propulsion of this particular type of cell. So again, the basic structure of a sperm is a head, a midpiece, and a tail. And in that head, we find the haploid nucleus, and we find the acrosome. The acrosome includes vesicles that are full of enzymes that are necessary for sperm penetration. This sperm will burrow its way into the egg using those enzymes to break down some of the proteins that are around the exterior of an egg. And then the midpiece has many mitochondria that generate ATP to power the flagellum, and the flagellum is the motor of the cell, so to speak. It allows for that swimming motion. This is what sperm look like under a compound light microscope. This is a particularly high magnification. Remember that sperm are the smallest cells in the body. Highlighted here are the 3 accessory sex glands of the male reproductive system, and collectively they play a role during arousal and ejaculation, and we'll talk about their individual roles in just a little bit. But the first gland that this moving semen will encounter along the ejaculatory pathway is the seminal vesicle. And again, this is a paired structure, right? There's a seminal vesicle over here as well as a seminal vesicle over here. And the next gland is the prostate gland, and that's singular. There's just one prostate gland that sits on the inferior aspect of the urinary bladder, between the urinary bladder and the penis. And then finally, along the ejaculatory pathway, we have the bulbourethral glands, and they're also paired. There's a left and a right. Both of them are shown here. So semen, this liquid that gets ejaculated, is a mixture of sperm and seminal fluid. So semen is a mixture of cells surrounded by fluid, and a typical volume of ejaculate is between 2 and 10 milliliters. And in each milliliter, a typical concentration of sperm is between 50 and 150 million sperm in each milliliter of semen. And the criterion by which male infertility is diagnosed is semen that contains fewer than 20 million sperm per milliliter. Seminal fluid is a rich mixture. It's a body fluid, so it's mostly water with a variety of dissolved solutes. And seminal fluid is produced by these 4 structures. The first one is the walls of the seminiferous tubules. So while the sperm are being produced, those Sertoli cells and other structures of the seminiferous tubules secrete fluid, and it's that fluid through which the sperm swim from the seminiferous tubules into the epididymis. And as those epididymis are propelled, as those sperm are propelled out of the epididymis and through the ductus deferens and the rest of the ejaculatory duct, those sperm are in a fluid, and at that point, at the very beginning of the ejaculatory pathway, that fluid was produced by the seminiferous tubules. And then along the way, fluid gets added to the semen by the seminal vesicles and the prostate. And as we'll see, the bulbourethral glands actually play a more important role during arousal than they do during ejaculation. So the seminal vesicles contribute about 60% of the volume of semen, and the prostate contributes about 25%. And the Cowper's glands, those bulbourethral glands, they are furthest along the ejaculatory pathway. They are closest to the penile or the spongy urethra. And they are most active during arousal, so prior to ejaculation. And they produce a secretion that's commonly known as pre-semen, or maybe you've heard of pre-cum. And this secretion, this is during arousal, so before ejaculation happens. This secretion helps to coat the mucosa of the urethra, so it coats the interior of the penile urethra. It has an alkaline pH to help neutralize or raise the pH that might be acidic due to any leftover residue from acidic urine that was left in the urethra. And seminal fluid produced by the variety of structures that we just talked about. Seminal fluid includes fructose. Fructose is a monosaccharide, and that fructose can be easily converted into glucose and then used by the sperm cells as a source of fuel for generating ATP. And I hope you remember that the midpiece of sperm include many, many mitochondria, and those mitochondria will use that fuel source to generate ATP, and that ATP goes to fuel or to power the moving flagella. Seminal fluid also includes antimicrobial enzymes. And so there are just microbes everywhere, bacteria and fungi. And there are bacteria and fungi inside the male urethra. There are bacteria and fungi inside the female reproductive tract. And so these antimicrobial enzymes are just general defenses. And then semen or seminal fluid has an alkaline pH. I talked about that a little bit in terms of the male urethra, where there might be some urine residue remaining, and that urine residue might have an acidic pH, so a pH below 7, and the alkaline pH of seminal fluid helps raise that as a protective function for the sperm. The sperm will be better off passing through a urethra that's lined with a fluid that has a more neutral pH. But also the sperm will be better off passing through a female reproductive system that has a more neutral pH. We've not yet talked about the female reproductive system, but the cervix and the walls of the vagina secrete fluid, mucus, that has a low pH, and this is a protective barrier. It inhibits bacterial growth, and it's also bad for sperm. And so sperm comes surrounded by this seminal fluid that has an alkaline pH to help make a healthier passageway for the sperm as the sperm travel through both the male urethra as well as the female reproductive tract. And then finally, seminal fluid includes clotting proteins. So semen outside the body over the course of several minutes, it will take on like this gelatinous type consistency. And this is because there are clotting proteins present. And the effects of clotting proteins vary across the animal spectrum. So in some organisms, the semen, once outside the body, these clotting proteins allow the semen to form what's known as a plug, to actually physically block, create a barricade inside the female reproductive tract. And this prevents any competing sperm from being successfully introduced into her reproductive tract. That doesn't happen in humans, but rather the clotting proteins are far weaker and just sort of allow the sperm to become this gelatinous, it's still a little kind of a runny liquid. It's just a thicker runny liquid. And perhaps this helps hold the sperm in the female reproductive tract a little longer, prevents it from so easily running out of her body. So prior to ejaculation, we're sort of working backwards here. Prior to ejaculation, there's erection. And erection is due to a change in blood flow. More blood flows into the penis than flows out of the penis. And the penis is really a hydrostatic pump. And so As blood flow changes, the erectile tissues in the penis become filled with blood or engorged with blood. And so earlier I mentioned these two erectile tissues, the corpus spongiosum and the corpora cavernosum. So corpora is the plural of corpus. So there's a single corpus spongiosum that surrounds the urethra in the penis. And then there are two corpus cavernosa or corpora cavernosa, these paired rods of erectile tissue that run the length of the penis. So in this image here, we can see both the corpus spongiosum and the corpora cavernosum. So maybe green is a good color to use here. We can see a little bit of the corpus spongiosum here, and we can trace the pathway of the penile urethra and even into the glans penis. This is a rod of erectile tissue. It's full of holes, and those holes become filled with blood during erection. And then the other erectile structures we see here, there's one rod here and one rod here. These are the corpora cavernosum. Again, a sponge-like tissue where the holes become filled with blood. And so to understand the physiology of erection, we need to understand these three erectile tissues along with blood flow into and out of the penis. So we're looking at a transverse section of the penis here, and we see our three rods of erectile tissue, starting with the corpus spongiosum. It's surrounding the penile urethra, and then finishing with these two corpora cavernosa, these two rods of erectile tissue running the length of the penis. And then we need to think about blood flow. So you remember that blood flows away from the heart in arteries, and blood flows back toward the heart in veins. And so highlighted here, we have some important arteries and veins. So an artery is going to allow blood to flow into the penis, and a vein will allow blood to drain out of the penis. And so during erection, we need to increase blood flow into the penis, allowing these erectile tissues to become engorged with blood. And then to maintain an erection, we need to decrease outflow of blood from the penis. So we're going to see some changes that occur with these blood vessels here that allow erection to happen and then allow erection to be maintained. So in order to trigger an erection, it requires an increase in parasympathetic stimulation. And that increase in parasympathetic stimulation is going to trigger the production of a very potent vasodilator called nitric oxide. And nitric oxide is produced in tissues all over our body, and it only acts locally, just in that neighboring area, in that vicinity. It will cause the blood vessels nearby to widen in diameter. And so in the male reproductive system, parasympathetic stimulation triggers the production of nitric oxide, and that causes the penile arterioles to vasodilate. And so this allows more blood to flow into the penis, and those erectile tissues become engorged or become filled with blood. So in this image, we see the sensory inputs that lead to the onset of erection. And the sensory inputs are shown here in these sort of gray-green boxes. There are a variety of sensory inputs that can lead to motor output that initiates an erection. And so these sensory inputs include mechanoreceptors or touch-sensitive cells located on the penis and other parts of the body, as well as higher brain centers, so your thoughts, your emotions, as well as visual input and olfactory input. So this variety of sensory inputs can generate motor output that looks like this. There's an increase in parasympathetic stimulation to the neurons of the penis, and those parasympathetic neurons generate or release nitric oxide, that vasodilator. And simultaneously, there's a decrease in sympathetic stimulation to the penis. And so sympathetic stimulation is dialed down, parasympathetic stimulation is dialed up. That means that nitric oxide is being released, and that has an effect on the diameter of arteries and arterioles serving the penis. They dilate. So this increases blood flow into the penis. That engorges those erectile tissues, which leads to the beginning of an erection. And an erection leads to, as we'll see in the next slide, we'll see an erection leads to the compression, pushing down on penile veins. And veins are responsible for draining blood away from the penis. So we have this increase in blood flow into the penis with a decrease in blood flow out of the penis. And so this arrow here is how an erection is actually maintained. In this diagram here, this very simple diagram here, we can compare the diameter of veins and arteries serving the penis. So in the state of a flaccid penis, meaning a penis that's not erect, the diameter of vessels collectively that allow blood to flow into the penis and allow blood to flow out of the penis, collectively, those vessels have equal diameter. So blood flow in equals blood flow out, and there's no erection. But during arousal, the penile arterioles vasodilate, as shown by that larger red oval, and the penile veins get compressed. So they have a narrow diameter as they're pushed upon by the penis itself to limit blood flow out of the penis. So here we're looking at these penile veins here. And during an erection, the erection itself sort of pushes on these veins to squish them pretty flat, limiting blood flow out of the penis. Perhaps you've heard of Viagra. Perhaps you get junk email on a regular basis. So Viagra is also known as sildenafil. That's the generic name. And Viagra is helpful for generating and maintaining erections in men for which that is a problem. And Viagra was initially studied in terms of the cardiovascular system. It was studied as a drug that might help patients who have heart disease because it can augment the effects of nitric oxide, a vasodilator. So if you're looking to ensure blood flow to the cardiac muscle tissue, a vasodilator might be helpful. But it was noticed when the drug came to clinical trials, a very noticeable side effect was priapism, the onset of erection. So men were taking this drug that was supposed to be for their heart disease, and they noticed that they got erections. And so the drug company Pfizer shifted gears and very successfully promoted this drug as a male enhancement to increase the capacity to maintain an erection. So now that you understand what makes an erection happen, what makes an erection go away? Well, all of the opposite things occur. So as there's a decrease in parasympathetic stimulation, there will be a decrease in the local release of nitric oxide. And under the influence of less nitric oxide, those penile arterioles will vasoconstrict. They get narrower in diameter. So now this is limiting blood flow into the penis. And so the penis begins to soften. And as that erection subsides, that's going to alleviate the compression on those penile veins. And so this will make it easier for more blood to flow out of the penis. And this is how the penis returns to its flaccid or non-erect state. So Viagra is one approach to treating erectile dysfunction, otherwise known as ED. And that's defined as the consistent inability of a man to sustain an erection long enough for sexual intercourse. So erectile dysfunction is not the once in a while, it's the consistent inability to sustain an erection long enough for sexual intercourse. And so it tends to be more common among older men, but it certainly doesn't mean that every man will experience erectile dysfunction. It's probable, but not inevitable. So what causes erectile dysfunction? The answer is lots of things. It's anything that could possibly disrupt the sequence of events that lead to erection. So there's a list of possibilities here. So erection requires a fully functioning nervous system and a fully functioning cardiovascular system. right? We require an increase in parasympathetic output, a decrease in sympathetic output, and blood vessels that are functioning properly to adjust blood flow into and out of the penis. Testosterone deficiency. Testosterone has an influence over libido, or how horny you are. Insufficient release of nitric oxide. If there's a problem with those nitric oxide producing cells in the penis, then sufficient vasodilation just won't happen. Certain drugs can inhibit the formation of an erection. Perhaps the most well-known is too much alcohol. So under the influence of alcohol, it's well known that men are not able to become erect or sustain an erection long enough for intercourse. And then there are a whole host of psychological factors. Your brain is your biggest sex organ, including anxiety and PTSD. And then there's just straight up tissue trauma. If the nerves or blood vessels or other surrounding tissues have been damaged, if there's enough local inflammation, then the nervous system and cardiovascular system changes required simply aren't possible. So in treating erectile dysfunction, the most important nugget is to try to get at the cause. If we can identify the cause of erectile dysfunction, that should lead to a more targeted, more effective treatment. So if a certain medication is causing ED, maybe we can change that medication. If low testosterone is causing ED, we can supplement testosterone. Viagra augments vasodilation. We can also deliver drugs locally through penile injection, and those drugs can cause vasodilation. We can also use a vacuum device that generates a low pressure system around the penis. And so you remember that fluids flow from an area of high pressure to an area of low pressure. By using this vacuum device, we can create a low pressure system around the penis, and that will cause more blood to flow into the penis. And then if anxiety or PTSD or some other psychological aspect is the cause, well then psychological therapy might be an effective treatment. So what happens during ejaculation? Ejaculation is defined as the forceful ejection of semen. And during ejaculation, the internal urethral sphincter closes. I hope you remember that there are two sphincters associated with the urethra. And during urination, both of those sphincters need to relax so that urine can pass from the urinary bladder into the urethra. But during ejaculation, we don't want semen to move into the urinary bladder. So the internal urethral sphincter closes, and that sphincter is located right around here. The internal urethral sphincter closes, and this prevents urine from being released during ejaculation. It also prevents semen from moving up into the urinary bladder. Ejaculation is the forceful ejection of semen. So let me remind you that sperm are produced here inside the seminiferous tubules of the testes. And then mature sperm are stored here in the epididymis. And during ejaculation, the sperm and seminal fluid are forcefully propelled along the rest of the ejaculatory pathway through the ductus deferens and through the various regions of the urethral, and finally out the external urethral orifice. So what propels the semen? And it's peristaltic contractions, so wave-like muscular contractions. There are smooth muscle cells that we find all along the ejaculatory pathway. And then we also find muscle within the penis. And collectively, these muscular contractions propel semen out of the body. And so this brings us to the end of the first segment of the reproductive system. This is slide 58, and we've finished our coverage of the male reproductive system. And in segment 2, we'll pick up with slide 59, and we'll talk about the female reproductive system. Thank you for watching.